let's get started. Really excited about the talk today. I think this is the most intriguing title that we've had for a Hutch at Home session so far. I know I am really curious about what we're going to learn today. So the thymus, the most important organ you've never heard of. And really delighted to have Dr. Dudakov here with us today as well. So Dr. Dudakov is an assistant professor in a program in immunology in the clinical research division at Fred Hutch, and also a professor of our integrated immunotherapy research center. I also want to add that he's been a great supporter of science education at Fred Hutch and is on the advisory board for our Frontiers in Cancer Research grant, and really appreciate that effort on his part to be part of education. So thank you in advance for that. And so I'm going to turn it over to Dr. Dudakov.
Thanks. All right, let me try and share my screen here. Should work. All right, I think you are sharing the wrong screen. Just a moment.
It looks good here.
As in, you can see.
Oh no! Now you're in presenter.
Now I'm in presenter. Right. Okay. It went the other way around. Just give me a sec. I'm sorry. Yeah, it doesn't want to do that one. That's okay.
This one is good. Yeah.
This one is good. Excellent. All right. Well, thanks for having me and inviting me to these. These are always very interesting. This whole new world of Zoom seminars. It's like I don't know. I don't often speak to my computer quite like this, and when you're in presenter mode like this, you actually just can't see anybody. So I hope there are still people there. So yeah, as Jen said, I'm going to talk about the thymus, which is, I think, to be a particularly interesting and important organ. I'm sort of going to take you through why that is, and then some of the work that we've actually been doing to understand its function, and particularly what we're interested in, which is kind of at the nexus of immunology and regenerative medicine, actually. And so we're looking to kind of regenerate this organ and how we do so. So I've sort of gone through a couple of different stories we're working on at the moment to really kind of get down to the science, but I've tried to kind of make it accessible. But if there are any questions or something's not clear, please feel free, just jump in and ask a question away. So I wanted to give a bit of a background about me first before kind of getting to the science, ease us into it. So as you can probably tell from my accent, I'm not from around these parts. So I come from a place in Australia called Melbourne, one of the biggest cities there, so the second biggest city. Lots of, culture, very, very beautiful city, actually, very, very similar to Seattle, actually. So in Melbourne, which is where I was born and grew up, it's slightly different to how it works in the States, in that often you don't necessarily go out too far, particularly if you're from one of the big cities. So I actually did my undergraduate at a university called Monash University, which is in Melbourne. It's one of the bigger universities in Australia. But, I actually started off quite, I don't know, I mean, I'm not sure if probably a lot of your students are like this, although in this country, people are a little more, focused, but I really didn't know what I wanted to do. And so in Australia, undergraduate education is much more career focused than it is here. And so, for instance, you can do law, you can do medicine, or, any of the vocationally, directed courses as an undergraduate. You don't have to do a, 4-year undergraduate bachelor degree first. So, I mean, I didn't know what I wanted to do or whether I wanted to do any of those sort of things. So I kind of designed my own incredibly broad course by doing something what's known as an arts degree, but also doing a science degree as well. So it was a dual double degree, arts and science. And actually I started off doing, as I've listed here, sort of more in psychology and philosophy and a few things like that. So I really didn't know what I wanted to do. Did a little bit of astrophysics in there as well. And then, I actually took a year off, went overseas, lived in Canada for a while, actually drove through all over the United States and through Mexico and a few places like that. And it was in that trip that I kind of, switched places with a friend. I was on that trip. So I was actually a little bit into filmmaking. I was into things like that. And he was doing some more hardcore sciences. He was into chemistry and biology and some of these biochemistry, things like that. We started speaking a lot more overseas. And I decided when I came back, I wanted to sort of get more into the kind of hard sciences. And so I went back, and I hadn't done any of these since middle school, essentially, because I'd been focusing more on maths and physics and some of these areas. And so I went back and did biology and chemistry and a few of these kind of very base level sort of subjects, and then really sort of switched course a little bit and started, one, on my arts degree, I got really interested in politics, and that's kind of my other love, although at the moment it's a little bit trying. But I was also getting into biochemistry, and then finally, and where I really settled down and kind of found my home, was when I did my first course in immunology, which was a course only offered to seniors. So at that point, these are 3-year bachelor degrees. And so I did immunology and really just fell in love and did very, very well in that class. And so was invited to kind of do a summer scholarship in a lab. And really that just led to everything else. So the summer scholarship led to being a research technician in the Boyd lab, this Professor Richard Boyd. And then from there, I did what's known as an honours year. This is a dedicated research year, usually as an entry to a graduate like Ph.D. or something like that. And that's kind of what I did. And I did all of this in Richard's lab in Melbourne, in Australia. So then, as is common for those of us in kind of the life sciences in particular, a lot of people are staying these days in Australia. We do very well there, but I really wanted to get off and explore the world. It's always been something I've been very passionate about, is travel and really seeing things. So when I finished my PhD, while I hung around Richard's lab just to finish a few things off, I then moved to New York, where I joined the lab of Marcel Vandenbrink at Memorial Sloan Kettering in New York. And this really actually fulfilled just a personal fantasy of mine to live in New York. I went there for the first time when I was 10 years old and kind of really just fell in love with the place. Returned, a few times over the years. And while I looked at many other places, so it wasn't sort of the only place I was focused on, it's really, it was a perfect happenstance that that's kind of where I ended up in the end. So I was a postdoc in New York for about 6 years. That's kind of the position you do outside, like once you finish your graduate school. But then also kind of transitioned into more of a senior scientist position in Marcel's lab. And so he is actually head of blood cancers at Sloan Kettering, essentially. So both the clinical and research side. So it's a very large lab. He's got a very large administrative staff as well. And so I was essentially the sort of manager on the scientific side and the lab side. So that was actually fantastic for me, because it really gave me an entry into things that we're just not trained in. So things that you really need to do when you start to run a lab, like personnel management and budget management, and things that are just not at all science at all, but you really need a good grasp on. And so that was really kind of invaluable to me. I had a pretty successful time there, both scientifically and not. Had both of my kids in New York, and then tried to strike out on my own. And that's pretty much where I ended up here at the Fred Hutch, where I started my lab in 2016. So this was just in sort of brighter times when we could all, gather around a table within 6 feet of each other. It was last summer, where, I've assembled a pretty great bunch of people, and I think we're doing some really, really interesting work. Although it's a very interesting field, there's a reason you've not heard of it, and there's a reason why it sounds like the most interesting title you've had. It's because nobody really thinks about it. And that can be really good, because it's my own little niche, it's my own little area, but sometimes it can feel like nobody really cares. So that's, the flip side of things, but that's all right. That's kind of academia for you. So that's kind of, my background and where I started and kind of where I've ended up. So we've been in Seattle the last 5 years. My wife and 2 kids, we've sort of settled down here. We've, bought a house, all that sort of thing, have a dog. It's very nice. So now to get to, some of the sort of science. So really what my lab is interested in, and this is very, very high level, is to say, we're just trying to understand the immune system, and in particular, how these particular cells in the immune system, T cells, develop. And I'll get down to that a little bit, you know, what they do and how they do it in a moment. But, you know, very broadly speaking, what happens is that all blood cells form in, does my cursor come through? I can't remember in these talks, hopefully it does. Anyway.
Yes, we see it.
Okay, excellent. So all blood formation occurs in the bone marrow. So all your red blood cells and most of your white blood cells and the different varieties of white blood cells form in the bone marrow. There is one exception to that, and that is your T cells. T cells require this very specialized independent organ for them to develop. And I'll get to some of those reasons in a second. And that's this organ known as the thymus. It's located just above the heart in the mediastinum region here. Incidentally, actually in a lot of cardiac surgery, particularly in kids, they actually have to remove some of it to actually get down to the heart. So it really, envelops the heart somewhat there. And so we're just trying to understand, you know, how T cells develop in the thymus and how this relates to disease. And then the sort of second part of that is to say, well, you know, all of this starts in the bone marrow. And so what are the events that occur in the bone marrow that kind of set the stage for that T cell development? Are there things that are occurring there that are required for what occurs later on in the thymus itself? But, you know, just to take a step back and just talk about T cells, because I think objectively they are the greatest, the coolest immune cell. I mean, there are others, there are immunologists who will fight me on that, but I agree, I think that's true. And the reason that is because of this really, well, both simple and complicated interaction. And so that's where on the surface of a T cell, they have this thing known as the TCR, and that's very creatively named as the T cell receptor. So it's a receptor very specific for T cells. No other cell has this receptor. And what the T cell receptor does, what the TCR does, is it recognizes what's known as a protein antigen or a peptide antigen. So that's just a tiny little bit of a protein that gets presented to the T cell. And it does that in the context of this other molecule known as MHC. And MHC is expressed on all of your cells, essentially. And so what it does is a lot of different peptides, a lot of different proteins are presented. But if the T cell recognizes it in the context of this MHC, it will set off a reaction. But the thing is, there are many, 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 many different antigens. So think of the number of proteins there are. So, you know, 30-odd thousand or thereabouts in a human body, and they can be chopped up into, you know, many different types of peptides. So 30,000 times, I don't know, some very large number. So it's a huge number of potential peptides that could actually fit within the grooves of these MHC. But the other thing is most of these are going to be your own proteins. Some of these are coming from, let's say, a virus or something like that, and that's something you want to be able to recognize, that's something you want to be able to target. So that's this green guy here. But, you know, most of them are just your own antigens, and you don't want to recognize those. And so if you do, that's where you lead to autoimmunity. And so a breakdown in what's called tolerance, that's the tolerance of these TCRs to not recognize your own self-peptides, leads to autoimmunity. So really what you want from the immune system is recognition of those foreign peptides, but no recognition of your own peptides. Now that's a really, really complicated problem to have. Very hard one to design, let alone for it to evolve. And I think that's pretty much the only way it's going to occur. And the reason, how that's basically been solved is to say, okay, well, what we're going to do is we are going to generate this enormous diversity in that T cell receptor. So every T cell receptor is a little bit different, you know, what it actually recognizes. So this guy here is not going to recognize this guy here. This one here is not going to recognize this one here. And so you don't get, you know, cross reactivity that way. And so if you've got this real diversity of T cells, you've got a greater probability of actually being able to recognize those peptide antigens. And then the second part, which I'll get to in a moment, is actually training those T cells not to recognize yourself. And so that's basically what the thymus does, is it generates this huge number of T cells. Each of them has a different receptor. Now, we actually don't know how many possible receptors there are. It's a very, very complicated mathematical question. It's probably somewhere in the order of 10 to the 18 to 10 to the 33 possible T cell receptors. Now, just to give some context to those numbers, because they always completely blow my mind apart, is 10 to the 18 is about the number of grains of sand there are on planet Earth. Now, that's the lower estimate of potential TCR, So the numbers we're talking about are absolutely phenomenally large. Excuse me. So the way it does this is it takes these stem cells from the bone marrow, and you really don't need to get too concerned about the different nomenclatures and things like that. We will not talk about any of these DNs, ETPs, things like that. Just know that there are these very discrete stepwise processes from a stem cell. This is a blood stem cell. This lives in the bone marrow. And then somewhere around about this point here, they enter the thymus, and then they go through these very specific stepwise differentiation steps, ultimately to come out with these T cells with a very diverse repertoire. And there are a bunch of steps in there. So firstly, as I said, they start in the bone marrow, they get into the thymus, but you know, these cells can become, they're stem cells, they can become lots of different cells of the blood system. So one of the first things they need to do is actually just commit to the T cell lineage, just commit to being able to become a T cell and not everything else. The other thing they need to do is expand. There is this huge expansion in the numbers of these cells. Very, very few cells come into the thymus. The thymus has a lot of cells in it. So in the order of 5 to 10 of these cells come into the thymus every day. And in a mouse, at least, the thymus is about 200 million cells. So those 10 cells essentially expand up to be, you know, many multitudes, many magnitudes higher. And then you've got these selection events. And I don't want to go into them in too much detail, but basically what these are 3 distinct points at which the thymus is teaching those cells, one, to recognize the MHC, because all of us kind of have different MHCs. And then second, not to recognize the self-peptides in the middle. So I often view it like a gauntlet, where if they see a peptide in the thymus, they're killed off basically. If they don't see anything, they're allowed to escape, and they're allowed to become just, you know, a normal T cell in the periphery. Now, this sounds simple because it's a very, you know, linear path. I know this is a circle, but it's a very linear path essentially from stem cell to a mature T cell. But the thing is, there's a reason why there is a requirement for this very, very specialized organ. And that is, you need the other cells in the organ to actually guide that process. So one of the other things about T cells is that they're very, very hard to make. You can often take a stem cell, smatter some what are known as cytokines on it, and generate a lot of different types of blood cells. You just can't do that with T cells. So they're very, very hard. They require this very specialized organ. And that's comprised of many different cells, primarily these epithelial cells, these TECs, thymic epithelial cells. And there are actually different flavors of them depending on where they are in the thymus, medullary epithelial cells or cortical epithelial cells, CTECs or MTECs. But then there are a bunch of other cells, dendritic cells, and we'll get to some of those a bit later on, fibroblasts, macrophages. We don't need to go into the roles of these too much, but just suffice it to say that all of these cells are involved in providing the instructive cues that drive this cell to become, you know, a mature T cell that is competent and restricted to yourself. That is, it's not going to recognize your own peptides, it's not going to lead to autoimmunity, and it could potentially recognize a foreign peptide. So I just want to talk about these thymic epithelial cells for a second, because they are kind of the hidden unsung heroes to T cell development. So as I mentioned, there are these 2 flavors, but they have kind of this really diverse role in, a diverse array of roles. So firstly, all of those things I kind of mentioned in the stages of T cell development are under the instruction of these epithelial cells. So for instance, the commitment to the T cell lineage, the selection, so that is the recognition of those T cell receptors to the MHC, both occurs by these cortical epithelial cells. And then in the medulla, you get these, so these MTECs, medullary thymic epithelial cells, this is actually probably one of the coolest things that the body can actually do. So what these MTECs are able to do is produce every single protein in your body. It doesn't matter if it's coming from your eye or from your little toe or from your heart or from your lung or from your bone. Every single protein is basically produced by these MTECs so that it can be presented to the thymocytes, the developing T cells, and this is what provides the gauntlet that I was talking about. So this is what provides the signals where if those T cells, as they're developing, recognize any of those antigens, they're deleted, they're killed off. We don't want that cell getting out into the circulation, because if it does and it comes across that antigen, it's going to lead to an autoimmune condition. And so these things combined lead to this diverse repertoire of T cells, and that allows a greater probability of actually being able to recognize, whether it be a vaccine or whether it be a pathogen, or something we're more interested in these days is cancers often have their own antigens and they can be recognized by T cells as well. But the thing is, this is not a static process, and the thymus degenerates progressively with age. And really, when we're talking age here, we're really not talking very old. So I'm not sure if there are any students on the call now, but you might think you're young, but in terms of your immune system, it probably doesn't think so. So the thymus starts involuting from around about puberty, and it starts just progressively declining. And these are just some images up here in the human thymus, where essentially what happens is you get accumulation of fat and you get loss of functional thymic tissue. The C relates to cortex and the M to medulla, as I was talking about before. And so you see just a lot more of these, fat regions essentially taking over. I don't want to go into this in too much detail, but just suffice to say that these things called TRECs are a way we can measure thymic function in patients. It's very hard to get thymic tissue. We just can't do it from live individuals. While you can do some imaging, it gets very expensive. It's very hard to measure thymic function, but we can measure cells that have recently emigrated from the thymus. And that's what these TRECs are. Don't worry too much about the name, it's a bit scientifically, but this is a way we can measure recent thymic immigrants, the cells coming out of the thymus, and that basically just progressively declines with age. Now, what this does is if you've got fewer T cells coming out of the thymus, so you've got fewer new T cell receptor repertoires, overtaking the old ones, you actually start to get kind of this expansion of pre-existing cells in the periphery. And so that is what are these called memory cells. They're cells that have essentially seen something before, and you want them there because you want them there as a response if you were to see it again. This is what provides the basis of vaccination, for instance. Without memory cells, we'd have no vaccination because you wouldn't have that recall to when you saw the pathogen itself. But the problem is we like to have a pretty even number of T cells out in the periphery. So if you're getting fewer T cells coming out of the thymus, there's a compensating mechanism where these memory cells expand. But because the memory cells have already seen something, we don't need lots and lots and lots of them. So if they start to overtake that repertoire, there's this sort of constriction in the naive repertoire. And so basically what that does is, so here is our kind of healthy, nice immune system I described before, where you get a multitude of breadth in that TCR repertoire. With age, you start to get a reduction. And so now, rather than having a TCR out there that can kind of recognize every pathogen and you have a high probability of being able to recognize something, now maybe you don't have this TCR. And so this, whether it be a cancer or whether it be an infection, slips through and we're just not able to deal with it. And so this can be a big problem. But you might ask, why is this a problem? So in otherwise perfectly healthy adults, it's really not. I mean, we can get by, obviously. I mean, there are some issues, and I'll get to those in a second, but on the whole, there's not that much of a problem. The problem is, though, is that there aren't too many perfectly healthy adults, and there's a lot of comorbidities, and there are a lot of issues involved where this can be a really big problem. And so one that I really like to highlight is to say vaccination. And so this is some CDC data of the efficacy of the flu vaccine. And it's stratified based on ages. And so you can see here that the efficacy in a younger pediatric cohort is a lot higher than even the 18 to sort of 64 bracket, and certainly more than the 65 plus bracket. So this is just the response to a normal year of flu vaccine. So usually you might get 60% in a young cohort. That might only be, 20, 30% in slightly older cohorts. So this can be a real problem. Obviously, we want, vaccines to be very efficient and work very well at the moment. So this could become quite a big problem, especially as we know that in our current, predicament, age is, a pretty big hazard for COVID and clinical responses. Something else I like to point out, and again, this looks a little more complicated than it is to say that, so cancer immunotherapy has emerged as a really important therapy against cancers, and in particular, one form which is known as checkpoint blockade. And really all you need to know about that is to say what we're doing is releasing the handbrake on T cells, because T cells will have, tumors will have mechanisms that they can suppress T cell responses. And so what checkpoint inhibition does is releases those suppressive responses. Now, what you can see by this chart, though, is to say that cancer immunotherapy and these checkpoint inhibition correlates really well with the mutational burden of the cancer, and that is the cancer's ability to produce antigens that can be recognized by T cells. And when I say recognized, I mean that they're not going to be recognized as yourself. And this is really key. And so these are called neoantigens. And a lot of cancers do generate, in fact, all cancers do. It's just some, take, uveal cancer, for instance, very, very low mutational burden. And so checkpoint inhibition works very, very poorly. Squamous cell works very, very well, and it's a very, very high mutational burden cancer. And the reason this is going to the TCR repertoire I was talking about before, because it's all about probabilities. It's all about there being a T cell there that can actually recognize that pathogen or neoantigen or vaccine antigen. If you don't have that, none of this is going to work. Even checkpoint inhibition like this is not going to work if you don't have a T cell clone there that can recognize that cancer. So another area this is a big problem is in the case of chemotherapy or bone marrow transplant. So both of these things are pretty synonymous in this instance, where these therapies are really damaging to the immune system. And so this is a graph of, after transplant, different cell types and their ability to come back. You don't need to know too much about the individual cell types and what they do. Just know that basically T cells, and in particular these CD4 T cells, one particular flavor of T cells, can take years to come back, if ever, and I'll show you some data on that in a moment. And so this can be a real problem, particularly in the case of transplant, where what you need to do to ensure a successful transplant, bone marrow transplant, is to wipe out the entire immune system and then transplant somebody else's, and then you're hoping for reconstitution of that entire immune system. If you don't have reconstitution of your T cells, you are basically going to be immunodeficient for quite some time. And this is the reason why recipients of bone marrow transplants need to be very careful about, where they go and who they're in contact with and things like that, because they are immunodeficient for a very, very long time. And this can actually lead to some really bad complications. So this is just a pie chart showing some of the effects of, some of the problems that can occur after bone marrow transplant. And these are sort of what will kill you after bone marrow transplant. So the biggest killer of all is relapse of the primary disease. So that is, the transplant just didn't work, essentially. Then you've got a disease known as graft versus host disease. This is where the graft itself can actually attack the body and see it as sort of foreign, kind of for the same reasons I was talking about before about those cells have not been trained for the recipient's peptides, essentially. Infection is also a very, very large problem. Now, we think that this lack of T cells can impact on all three of these really large killers after transplant. Now, the reason why this is because the thymus is actually really, really sensitive to insult, very, very sensitive to damage. And so what I'm showing you here, so this is in a mouse model showing 4 different types of damage, essentially. So we've got cyclophosphamide up here. This is a very commonly used chemotherapy. Dexamethasone, which is a corticosteroid, it's actually also used very commonly for immunosuppression, has actually emerged as a very promising COVID-19 therapy actually as well. TBI, which I'm going to come back to, so remember these letters, but this is total body irradiation. This is where you actually irradiate a mouse, give gamma radiation to an animal. This is often used in the transplant setting, so it is used clinically. And then LPS, which is lipopolysaccharide, which is a part of the bacterial cell wall essentially, and just mimics a bacterial infection. And so with all of these models, you can see this is total thymic cellularity. This is the number of cells in the thymus just drops down precipitously within the first couple of days after each of these damages. But the thing is, it also has this really remarkable capacity to regenerate itself. And so this is very, very similar. So this is just total thymic cellularity. This is after chemotherapy, that cyclophosphamide that I mentioned. And you see this massive drop off in the number of cells, but it actually comes back pretty quickly. And this is actually in the human setting. This is data from several years ago, again, using chemotherapy. This time we're just looking at T cells. Well, this is not our work actually, but looking at CD4 T cells, this particular flavor of T cells out in the periphery. And again, you see a very, very similar trend. You get this real drop off, drops down to almost nothing. This is chemotherapy regime 1 and regime 2. And then you get a slow, recovery afterwards. So by around, 10 to 15 months after, they're more or less recovered. Now, very curiously, we've actually known about this regenerative function of the thymus for far longer than we've known what the thymus actually does. So these are papers that I've pulled out from 1924 and 1948, showing the regenerative capacity of the thymus after basically superanelectomy, sorry, it's a terrible name to say, where basically they're taking out the adrenal glands more or less. And you get this regeneration of the thymus. Now, it wasn't actually until 1961 that we actually learned that the thymus was the primary site for the generation of T cells. And I do like to point this out at the moment. So Jacques Miller, who is still alive and still actually goes in to work quite often at the Walter and Eliza Hall Institute in Melbourne, where I'm from, actually discovered the function of the thymus. And he's probably, I think, the only person, in the last, God knows, several hundred years who could actually say, I discovered the role of an organ. He actually won the Lasker Prize last year, which is one of these sort of, pre-Nobels, a very, very prestigious award. But the thing is, this regeneration, even though it's got this really, really remarkable capacity to regenerate, that capacity decreases with age. And so this is what I was talking about before, where the thymus ages, the actual ability to recover also ages and also declines over time. And so this is in a mouse setting where we've got just normal, this is what I showed you before in the blue, in a normal, what we call a young mouse, 2-month-old mouse. In the red is a 9-month, a middle-aged mouse, and it takes a lot longer to recover. Now, this is the same data I showed you before, the same paper I showed you before, only this is a 23-year-old patient. And even after almost 2 years, they still have not recovered their CD4 T cell count. So it can take years, and some people just never get them back. And a 23-year-old is pretty much nobody's definition of an old person. So we're really talking, when we sort of talk about age and aging immunity, we are talking quite young here. So the mechanisms underlying this regenerative process, what we call endogenous thymic regeneration, that is natural thymic regeneration, are really poorly understood. And so this is pretty much a central question of my lab, is to really understand how does this tissue regenerate itself. And really what we're trying to do there is to say, we think one approach to developing therapeutic strategies for boosting immune function. And so that could be to improve responses to pathogens or vaccines or immunotherapy, as I mentioned, kind of in the settings of age or injury or infection or cancer. And so we actually think what we're working on is quite broad in its applicability. And we can actually hopefully improve quite a few clinical outcomes that way. So that was a very broad sort of description of kind of what the thymus is and what it does and kind of some of the problems. And now I kind of want to take you through sort of 2 stories that we've been working on, and one of which we just submitted late last week for publication. So hopefully that goes well. And the other will hopefully be submitted in the next couple of weeks. And these are some of the mechanisms that actually underpin this process of regeneration. And so how do we do what we do? Well, firstly, we work pretty much exclusively in mouse models, although we're starting to get into some clinical samples as well. And so with that, we often use a kind of bread and butter technique to damage the thymus. That's total body radiation. So we take a whole mouse and we basically give the whole mouse a radiation. And you see this degeneration in the thymus and then regeneration. And we try and understand that. And we understand that by looking at, the composition of cells within that. And we've got several ways we can do that, either with imaging or flow cytometry, for instance. We do a lot of molecular biology, so looking at gene expression and things like that, RNA sequencing. This is a new way of doing these large scale gene expression changes. We do a lot of modeling, so that is to take, let's say, knockout mice or different mice with different genetic modulations to see what that may do, whether that perturbs or whether that improves regeneration, and see what goes on in there and try and tease it apart. And then we also really ultimately trying to look at T cell function. That is, if we develop any therapies, can we actually give it to mice, and can we then, see any responses in T cell function? So I just point this up because it's very, very cool, but that is, something we've been concentrating on quite a lot in the last couple of years is the vasculature in the thymus. That is the endothelial cells. And so you can see here, this is some whole organ imaging. It's called light sheet fluorescence microscopy. Excuse me. And you can see all of the little branches and vessels within one kind of untreated thymus. It's very, very cool. But a lot of this is sort of underpinned by the fact of these two pathways that over the last few years we've identified as being able to mediate this process of endogenous thymic regeneration. And so these are based on these two molecules, and I'm going to come back to them quite a lot, being BMP4 and IL-22. And so what happens is, in the first of these pathways that we published a few years ago, this is when I was a fellow in New York, is that damage to the thymus triggers these cells known as dendritic cells to produce this molecule known as interleukin-23. And this is what we're going to concentrate a lot on a little bit later, so take note of the IL-23. That then initiates a triggering within another cell known as an innate lymphoid cell to produce interleukin-22. And then that stimulates the thymic epithelial cells I was talking about before to then lead to regeneration. So that's one pathway. And then a couple of years ago, we uncovered another. And this one, again, thymic damage, in this case, triggered endothelial cells to produce BMP4. And then again, BMP4 targets the thymic epithelial cells and mediates regeneration. Now, we did this using a bunch of different things. These two papers are quite beastly kind of papers. We used different knockout mice, that is mice that don't have IL-22 or IL-23 or BMP4. We did a lot of bioinformatics approaches, so looking at, back then microarray data, but also RNA sequencing data and things of that nature. And I'll show you some of these later on, is we do a lot of molecular analysis to really try and tease apart the kind of functional anatomy of what's going on at the molecular basis. So the question we've really been trying to answer is to say that we've got these distinct pathways of regeneration coming from different cells. They both target these thymic epithelial cells, but they're both triggered by the damage itself. So we wanted to know, is there a common mechanism that actually triggers this tissue regeneration? And so we like to think of this as, is there a damage sensing trigger to this regeneration? And so we really focused a lot on these two cell populations, endothelial cells that produce BMP4 and dendritic cells that produce IL-23. And so we're looking for things that might impact on both cells and their ability to produce these factors. Now, going back, when we first described our IL-22 story, something we noticed was quite interesting, and that is, if you look at thymic cellularity, so this is again just looking at the total number of cells in the thymus after giving a dose of sublethal total body radiation. So this is a radiation that's not going to kill the mouse, they're going to recover, but it is going to be quite damaging to their immune system, damaging to their thymus. And you can see that here, the cell number just declines precipitously, and then slowly comes back by about 8 weeks or so, it's back to normal. if we look at the levels of IL-22 or BMP4, the yellow over here or the blue over here, what we see is that while it doesn't change sort of initially in the first couple of days, it comes up pretty quickly and then comes back down pretty quickly. And it does that almost to the inverse to the nadir of thymic cellularity. So when thymic cellularity gets to its lowest, that's when the sort of triggering goes off. Now, something else we noticed, and I don't want this to be too complicated, but this was using a bunch of different mice with deletions for certain molecules. And in immunology, we're very fortunate because if we knock out certain molecules, we can block the development of these T cells. So take these three knockouts here, IL-7, IL-7 receptor alpha, or RAG1. They are all blocked, they are all blocked at this stage between this DN to the pre-DP double negative to the pre-double positive stage. Similarly, if we knock out this T cell receptor beta, you don't get these double positive cells and so on and so forth. Now, what we noticed here was really, really interesting, and that is if we took these mice and just measured the amount of IL-23, we actually saw a really large increase in mice that didn't have these double positive cells. So that is in the IL-7 receptors, the RAGs, the IL-7s, and the TCR betas. But if they had double positives and didn't have any of the other cells, there didn't really seem to be much of a change. So the way we interpreted that was to say that what we thought might be going on is that, is there a signal coming from those double positive cells, this particular stage of thymocyte development, that actually blocks the production of these regenerative factors, in particular IL-23 and BP4, but after damage, or in those genetic models I mentioned before, where you get rid of those double positive cells, now the handbrake is released, and now you get the production of these regenerative factors. So we started wondering what could actually be mediating that process, and something that double positive cells really, really like to do is die. And the reason that is for those selection events that I was talking about before. So when the thymus is teaching the T cells to develop and teaching the T cells not to recognize your own peptides, most of the cells are killed off. At this stage of the double positives, around about 99% of the cells do not go through this stage. And the reason that is it's kind of called the Goldilocks zone. This is what's known as affinity, so that is kind of the strength of the reaction between the T cell receptor and the MHC. If it's too low, the cells are killed off. It's not good enough. If it's too high, also killed off, not good enough. Will also lead to autoimmunity if it's too high in affinity. So it's really just in this Goldilocks zone in the middle where it's just right that you kind of get survival, and really only around about 1 to 2% of these cells actually does make it through that stage.
Can you quickly review double positive?
Oh, I'm sorry. So yeah, I didn't want to get too bogged down in that. It is a type of developing T cell. It's actually the most numerous thymocyte, numerous developing T cell in the thymus. I don't want to get too bogged down because it can really go, you can really
That's
get,
enough
please
for us
ask, I'm happy to, but it's just, you can really go quite deep in this. What happens at this stage is that they start to express the T cell receptor.
Yeah.
So what it's trying to do at this point is to say, because these are just random events that that generate these T cell receptors. So it's first trying to say is, can I bind to MHC? The peptide, the protein in this point probably isn't that important, probably isn't that important. It's really, can it bind to the MHC? And if it can, then it can go on and do its job later on. And so, like I say, in the normal thymus, it's probably around 80 to 85% of the thymocytes are double positive cells, so they're clearly the most numerous cell type, but 99% of those are killed off because they either don't recognize MHC at all, or they recognize it too strongly.
Okay, that's good. Thank you.
So we hypothesized that this apoptosis, this act of dying by these cells via this process known as positive selection, that's what I just described then, might be mediating this suppression of the production of these factors. Now, I want to just stop for a moment and just talk about a particular assay that we're going to use a little bit. And I've sort of focused on this because we use this several times to try and mechanistically tease these things apart. And so what we're able to do is we can take endothelial cells, ECs, or dendritic cells from just a normal mouse, and we can put them in culture. And then we can stimulate them or modulate them in culture and then just look at their production of IL-23 or BMP4. And that's what we're going to do kind of repeatedly. In this particular case, we're trying to ask the question, do dying cells, and there's another term for dying cells, which is called apoptosis, which is a programmed form of cell death. This is the way that most cells die. Do apoptotic thymocytes suppress the production of these factors? So what we did here is we took dendritic cells or endothelial cells and put them in culture. And then we took from the thymus a bunch of thymocytes, and then we either induced apoptosis using actually dexamethasone, which I mentioned before, corticosteroid, or inhibited apoptosis using this drug known as ZVAD. Doesn't matter what it does or what it stands for, but it basically inhibits apoptosis. So now we're putting onto these endothelial cells or dendritic cells, cells that are dying or cells that are not dying. And then we're going to measure the response afterwards in either the production of IL-23 or the production of BMP4. And so pretty much in both instances, so the blue here are where they're being incubated with dying cells, the sort of the sort of browny green are when they're inhibited they don't have apoptotic cells. And so what you can see is that in both endothelial cells, and this is BMP4 expression measured by qPCR, or in dendritic cells, IL-23 expression, this is measured by flow cytometry, we actually see a large increase in the production when we put on cells that are not dying compared to those cells that are dying. So this really says that the apoptotic thymocytes, the apoptotic cells, are suppressive to the production of these factors. So we want to know a little bit more about how apoptotic cells are actually detected. And so there's a rich body of literature on this at the moment, and I don't want to go too much detail other than to say that there are these things known as TAM receptors. This stands for 3 different types of receptors, tyro, axle, or mer. That's just the name of the receptor. And basically what they do is they bind through these intermediaries, either Gas6 or PROS1, this molecule known as phosphatidylserine. Now, phosphatidylserine, or PS, just because I don't want to have to say that many times, is a typical component of the cell membrane. So usually it sits on the inner cell membrane. I don't even know what it does actually during normal times, but it's one of the first things to occur when a cell starts dying via apoptosis, is it inverts from the inner cell membrane to the outer cell membrane. Now, when that happens, that can then start to get detected by these TAM receptors and lead to, a response. So these TAM receptors detect apoptotic cells via this, very early process in apoptosis known as PS inversion, phosphatidylserine inversion. So we wanted to know if that was what was going on here. So we did this same assay that I just described before. So again, we induced apoptosis, only this time we didn't inhibit apoptosis itself. We inhibited the detection of apoptotic cells by using a drug known as RxDX, which blocks that TAM receptor interaction. So again, it's not inhibiting the apoptosis. We've still got dying cells in here. We're just inhibiting the interaction between those dying cells and our target cells, the dendritic cells and the endothelial cells. And much the same way, so again, so grey in this case, sorry, the colours aren't consistent, obviously. Grey is with apoptotic cells, the blue is with the inhibition of the detection of these cells, and we see this really large increase in the ability to produce these factors. So it really seems to be that the detection of apoptotic cells seems to be suppressive. Now, there's actually a whole bunch more, and I don't want to go into a lot of this because it gets quite molecularly dense. But suffice to say that what we think is going on is that in the normal steady state homeostatic thymus, when there is a lot of apoptosis going on, we get detection via these TAM receptors. That leads to this downstream molecular signaling cascade that basically suppresses the production of BMP4 or IL-23. But after damage, when those cells are removed by depletion, essentially, those cells just die off, it's essentially a dead man switch, because now the handbrake is released. You don't get activation of those TAM receptors. You don't get activation of the downstream molecular cascade, and now you get the production of these factors. They can act on the thymic epithelial cells, and you get regeneration from there. Excuse me. So I realize I probably don't have that much time, so I might actually finish there because I won't go into the second story. So, because I don't think I've got time for it. So basically what we've found is, I think, a pretty unique global trigger for the regenerative process. And we actually think there might be a flip side too, where there are kind of different forms of cell death, where this is a suppressive form, but we actually think we might have a more of a promoting form in another form of cell death after damage. And so I'll just go to, so what we're trying to do here is to say, can we take these therapies, can we take these mechanisms that we've identified and generate new novel therapies for boosting thymic function? And we've actually done that in a few of our different ways, and this is something we're working on quite a lot at the moment. But, you know, where would we want to actually do this? And so I've just listed here 4 different instances where I think this could actually be really beneficial. So bone marrow transplant, obviously this is what I mentioned before. So just the sheer recovery of those T cells after transplant is incredibly important for clinical outcomes. Just recipients of chemotherapy in general, because most chemotherapies are actually really quite suppressive to immune function, and it takes a long time to recover. So I think there could also be a lot of scope there. And then the flip sides here are vaccination immunotherapy, because if we're able to regenerate the aged thymus, the non-acute damaged thymus, we can probably really improve responses to things like vaccination immunotherapy. I mean, 60% for a flu vaccine, even in the youngest health, is not very effective. So maybe things like that can actually be improved by improving our thymic output. And so with that, I'd just like to sort of point out a few of the people in the lab who did this work. In particular, of the story I sort of presented is Sinead Kinsella, who's a staff scientist in the lab, who's just been really remarkable at teasing apart this molecular pathway. And here's us this year actually now having a socially distanced picnic, which was lovely to actually see everyone in person rather than just a Zoom call. So thank you very much for your attention. If you've got any questions, I'd love to take them, or comments or anything else. Thank you very much.
Yeah. Thank you so much. Go ahead and put your questions into the chat, or feel free to unmute yourself if you'd like. As we're switching over here, I was kind of wondering, and I asked this as a person who, you know, I was kind of shocked by the pictures of the aging thymus, I must admit. And so is there a possibility to use some of the knowledge from regeneration to support thymic function in people who are getting older?
That is certainly our hope. I'm not sure that it would be, this is what we've got to figure out is, you know, how does that work? Is it something that just everyone takes at a, you know, ad infinitum, or is it something you do in a more targeted approach, let's say for a vaccine or something like that? But yeah, this is exactly where we want to go, because we think, and we're sort of getting into some of the clinical studies to try and demonstrate this, but that one of the reasons why some of these therapies fail is because of thymic function. And so that still needs to kind of be formally shown. But I think if we were to do that, I think there is a really solid rationale. And I think it's certainly the second thing we look at. We start with the damage models, and then we always plug it into an aging model to see whether we can get improved responses. So absolutely, definitely.
We have a question in the chat. What is important to know about COVID-19 vaccines and the thymus generation of memory T cells?
So it's a really excellent question, and one I've got a couple of answers. We don't know a lot is probably number one, because, you know, we just don't know a lot in general at the moment. So the thymus itself doesn't directly generate memory cells, but what it does is generates the cells that then become memory cells. So the way you become a memory cell, which is obviously going to be incredibly important for vaccine strategies, is, this goes back to one of my comments earlier on, you have to have a T cell clone there to begin with that can recognize that vaccine or pathogen or things like that. If you don't have that, you're not going to generate memory. And so I wonder if, you know, one of the reasons why we see poorer prognosis in aged people, poorer prognosis in men as well, and it's something I didn't touch on, but excuse me, testosterone is incredibly suppressive to thymic function. And we know this. And so, you know, one of the reasons aged immune system is not the same in both men and women. And so one of the reasons why it might be worse could be some of these kind of factors. And so, you know, it's an excellent question and one we don't know a huge amount on. I strongly suspect there is a link there, and it's a link that not many people would be looking at, you know, at all, essentially, except for us and a few others in our field. Yeah.
There was a point where you were talking about the MTEC cells producing every protein that a human body could make.
Yeah.
How does that happen?
It's really impressive, and we don't know the half of it, but, you know, so we know of a few. So there's one particular transcription factor known as AIRE, A-I-R-E, and stands for autoimmune regulator. And essentially what this does, although, like I say, we're still trying to figure this out, is it can transcribe all these different proteins. Now, when I say it like that, it sounds probably worse. It's not like the whole protein is there. It's like kind of only bits of it.
Yeah.
Yeah, exactly, because that's all that needs to be presented. But this is what we're trying to figure out. But it's never like it's a fully mature, you know, capable protein that is generated. But there is kind of that capacity to do so because, you know, they can do this. It's really remarkable. And as I say, we know they can do them all. AIRE is only part of the story. It can only transcribe, you know, a small fraction, 5,000 or 6,000 or something like that. So we know there's more out there. We just don't know what they are. So it's still a very big open question. Yeah.
Lots of stuff for students on the call to research in the future. Thank you so much. I wanted to, before we close out, share with everybody that we're going to change the Hutch at Home schedule in the school year and go to once a month. And these are the dates that I'm thinking of right now. And I'll be sure to communicate with everybody when these are coming up. We're going to also once a month have a virtual field trip. So similar to one of the talks that we had earlier this year, that'll include a visit to a lab and talking with some people in the lab as part of that. So we're changing up the format, but I hope that we will still be able to see you. And if you have a moment, feel free to unmute yourself and put some reactions with your photo, and you can feel free to turn your video on if you like, or just put something into the chat to say thank you to Dr. Dudakov for joining us today. And I put the link for feedback into the chat as well. And thanks everybody. I really appreciate you coming today. And I especially want to thank again, Dr. Dudakov for educating us about the thymus.
Thank you.
Thank you.